So our first lecture kind of dealt largely with the breakdown of endogenous sources of proteins, and an important kind of component of maintaining normal cellular function is recycling old proteins and enzymes and so forth that are not functioning well. So we talked about that aspect. On our second lecture, we kind of talked a little bit more about the chemistry that is involved in the urea cycle and the disposal of nitrogen, and then and the disposal of the alpha keto acid skeletons that are remaining. And so in our last section here, we're only going to spend probably about a half a lecture on this. We're going to talk about other places that you utilize nitrogen in your body. Well, one of the places is that you actually need to think about building your own amino acids. There are some amino acids that are what we call non-essential, meaning you can actually make them in your diet or make them in your body, so you don't need to consume them from your diet. So the question is how do you do that? Well, you actually reverse a lot of the processes that we've talked about, but you need to have a source for nitrogen, and the source for that nitrogen is often glutamates and nucleotides. We'll get to nucleotide metabolism in our next chapter, but glutamates are an important source of nitrogen, and that's why things like MSG, which stands for monosodium glutamate, that's a flavoring agent. It's thought of as kind of the fifth sense. Why you actually have a taste for MSG is because it's an important building block for you to make your own amino acids. Heme. We've seen hemes a lot, whether it was in hemoglobin, whether we've seen heme or heme related species in electron transport, in photosynthesis. So building these hemes is important, and as we kind of see over here, we actually utilize some amino acids like glycine that help to provide our nitrogen atom building blocks. And so lots of this chemistry happens in red blood cells in the liver because there's lots of needs for heme, for cytochromes P450 enzyme activity. So we'll spend a little bit more time talking about this or reviewing some of the concepts that we've already discussed in class. But not only do we need to think about synthesizing heme, we need to think about how we degrade heme. So again, hemes are a significant source of nitrogen in your body, but bruises, right? We've got this extracellular degradation of red blood cells basically is going to take this heme, and we need to break it down into some components that we can excrete because heme is very water insoluble. So what happens is we do some oxidative chemistry here to open this up and generate biliverdin, bilirubin, urobilinogen, stercobilin, and urobilin. So not names that you need to remember. This is one that we're going to talk about, bilirubin. Hopefully that's one that you've heard about. If not, we'll kind of talk about it on the next slide. But not surprisingly, these are brown colored and yellow colored because how you dispose of these, stercobilin and urobilin, is in urine and then in fecal matter. And so again, those routes are an important way to get rid of heme garbage, if you will, that's in your body. So again, an inability to get rid of this garbage often happens and stops at this bilirubin step because you need kind of microbial enzymes to get rid of that. So infants are very susceptible to jaundice, particularly breastfed infants, because they may not be going through effective digestion. And so they're going to have a lot of buildup of bilirubin. And so this is yellow colored and it gets deposited in excess in the skin, whites of the eyes, and so forth. It's also commonly seen in an older population, particularly those who might have liver issues. Cirrhosis of the liver generally leads to jaundice, and that's because of the inability for the liver to do its initial chemistry to facilitate this process. A couple of other uses for nitrogen that we'll talk about is the production of nitric oxide. And so nitric oxide synthase is going to convert arginine into a couple of intermediates here. But importantly, it's going to generate nitric oxide. And nitric oxide has a lot of important biological implications. It's an important vasodilator, which is why it's useful in the production of NO with Viagra. It's an important component of the central nervous system and kind of nerve impulse transmission. It's also a potent cytotoxin. And so your body, even though we've talked about superoxide radicals being very damaging, your body has the ability to kind of harness the activity of those and use them for good purposes. So the last thing that we'll kind of do a little bit of time talking about and use it kind of as an opportunity to review is to remind ourselves of the prominent elements that we've talked about in our metabolic species here. Carbon, hydrogen, oxygen, obviously very important. We focused on those with our lipid and carbohydrate metabolisms. Then we've talked about nitrogen that kind of came into play with protein metabolism. We'll talk about phosphorus a little bit more as we talk about nucleic acid chemistry in our next chapter. But what's really interesting to think about is kind of the major forms of carbon and nitrogen in the biosphere are tied up in very unreactive molecules, carbon dioxide and nitrogen. And so part of the solution, and we've already talked about this in our photosynthesis chapter, is to talk about how carbon dioxide is a source of carbon, and we fix carbon as carbohydrates in photosynthetic plants via the Calvin cycle. So again, Rubisco being arguably one of the most important enzymes to facilitate that process. But we also need to think about how we can fix nitrogen. So most plants are not able to fix nitrogen on their own, and so we have symbiotic bacteria called diazotrophs that are found in kind of these root nodules of legumes. They help to fix nitrogen, and essentially they take this very unreactive N2 and convert it into a very bioavailable NH3. So we'll kind of talk about a little bit about the chemistry for how this happens, but again, this step is hugely important. How do you take this highly unreactive N2 and convert it into the very bioavailable N3? So again, reduction chemistry is going on here. But really important chemistry. Maybe you guys know, if you've kind of talked about organic chemistry reactions, you want to do something in an inert atmosphere, you do it under nitrogen because it's so unreactive. So we'll spend a little bit of time looking at the structure and activity of nitrogenase. Just like Rubisco, it's arguably probably one of the more important enzymes that we think about in terms of making bioavailable elemental nitrogen and carbon. We'll kind of show an application of this when we think about crop rotation. You've probably heard about crop rotation. The idea is putting plants like alfalfa and so forth allow you to replenish the nitrogen in the soil, so you don't need to apply fertilizers. But again, fertilizers are another way to just add nitrogen into the soil. Other sources, actually lightning and electricity is an important mechanism for fixing nitrogen. Composting, right, is if we provide kind of that source of nitrogen through the breakdown of other fuels. Genetic engineering, unfortunately, has not been as successful as it has with other things in terms of trying to facilitate the reintroduction of nitrogen as a bioavailable form. So some interesting things to kind of think about, some other uses and aspects of nitrogen in biochemistry. We'll get to more when we talk about our chapter on nucleic acid chemistry.